Welcome to another Harley Healthcast Inside 108. My name is Simon Marsh. And with me today, I've got Dr James MacKay, who's one of those rare breeds. He's called a genetic oncologist, which means he's a cancer doctor who knows more about genes than most people. So what I thought we'd do, start with James, just take us through how genes work and how it can affect disease.
So, when we examine different genes and different cancers, we're looking to find if we can identify a fault in any of the genes. And if we do identify a fault, say, in one of the genes, that suggests that that gene is not working properly. And the result of that is that it increases the risk of a specific cancer developing. So they are essentially what we call tumour suppressor genes, which really means that if they're working properly, they suppress the tumour. And if they're not working properly, they increase risk of that tumour developing.
Okay, so suppressor genes will damp down things from developing into cancers?
Yes.
Yeah, okay.
If they're working properly.
Right, okay. Now, let's take that back a step. What are genes?
So, genes are essentially a lineup of letters. There's only 4 types of letter in the alphabet: C, G, T, and A. The BRCA1 gene, which is for breast cancer 1 gene, is made up of 100,000 letters arranged in a sequence. And a faulty gene is caused by a change of a single letter. So it's a subtle spelling mistake, and it can be anywhere along the whole lineup of the gene.
Gosh, okay, so that can be quite difficult to find, I'd imagine. You mentioned tumour suppressor genes. Are there, that damp down things, are there genes that, if they don't work properly, will make things happen more quickly?
Yes. So if a tumour suppressor gene is not working properly, then that increases the risk of that cancer developing. But it also increases the risk of the cancer developing at a younger age.
Right, okay.
So in individuals with a faulty gene, the chance of them developing cancer at a younger age is increased.
Right. And how many genes have we got?
30,000 all in all.
Okay.
But there's only a very limited number of cancer genes.
Yes.
In that group.
That we know about so far.
That we know about so far.
And it's interesting, in the many years that you've worked with here at 108, the number of genes we've come across related to breast cancer and cancer is going up, isn't it? We're finding more all the time.
Yes. There's a major research effort being conducted across the world, actually, looking for new genes involved in different cancers. And as these research efforts mature, we end up being able to bring these genes into clinical care, which is what we now have. So we now have panels of different genes in different cancers available to be used in clinical care, which is what I do.
Yeah, and so you've been sort of in this right from the start, when we only really knew about the 2 breast cancer genes, BRCA1 and 2. So what's the biggest change you've seen in genetic oncology over the last decade or even 2? What really changed?
So I think there's 2 major issues. First issue is that we're able to do the testing much more quickly and more accurately. So initially, early on when I started, we ended up with a lot of inconclusive results, which actually is really difficult to explain to people. As our testing improves, we're now much more accurate and we get many less inconclusive results, although we still do occasionally get some. And I warn anyone that's thinking about having testing that it is possible that we find an inconclusive result.
So it's much easier to test people now. Can you test anybody?
Yes.
Okay. So what we've learned then is that we've all got 30,000 genes, some of which can go wrong and help cause cancer at a younger age. But we can start testing for these now. And what I'd like to, James, is get you back at some point. We go through more detail about the different sorts of genes we can test for and how that might help people perhaps develop strategies for detecting breast cancer early. James, thank you very much. We'll get you back very soon.